In this talk, we will learn how the epidermis forms from the ectoderm and different ectodermal appendages that arise from the epidermis.
The skin is the largest organ of the body, and it is made up of three major layers. The first one is called as the stratified epidermis. So when we look at the figure that is shown to the right, the epidermis is labeled in the blue box, and it is the topmost layer of the skin. Below the epidermis, we have the underlying dermis, and that is shown also in the figure in the blue box. And finally, we have melanocytes that are derived from the neural crest, and this is where we can see also in the blue box. The melanocytes are the cells that actually will make the pigment, which gives the color to our skin cells. The epidermis is induced from the surface ectoderm to the actions of BMP. Now, if you remember from previous chapters' information, the surface ectoderm has the propensity, or its default state is to develop into neural tissue. It is the actions of BMP that actually inhibits neural specification of the surface ectoderm and makes it to become epidermis. Now, during early development, the epidermis is only one layer thick, but as development progresses, it forms two layers. One of these layers, which is the outer layer, forms what's called as the periderm. Now, the periderm doesn't last very long and it sheds once the inner layer differentiates. Now, the inner layer is called as the basal layer, or it's also called as the stratum germinativum, and this is the part that will actually undergo differentiation to form the epidermis of the skin. Now, epidermal stem cells are present in the inner layer in the basal portion, and they can undergo either symmetric or asymmetric cell division. When they do undergo asymmetric cell division, the daughter cell that remains attached to the basal lamina remains as the stem cell, while the daughter cell that leaves the basal lamina will undergo differentiation and ultimately form the skin cells, which are the keratinocytes. Now, differentiation of the epidermal stem cells is mediated by Notch. So we have to remember the role of two paracrine factors, BMP and Notch, when it comes to the formation of the epidermis. So as I mentioned in the previous slide, Notch plays an important role in the differentiation of stem cells into keratinocytes, which are the differentiated epithelial cells. The new cells are formed from the stem cells in the basal layer, and as they undergo differentiation, they push the old cells to the surface. So if we look at the figure that is shown to the right, in the blue box, we can see cells that are present, and these are the stem cells which are attached to the basal layer. As these cells undergo cell division, and we can actually see two such cells undergoing cell division, the daughter cell that maintains contact with the basal lamina remains as a stem cell, while the daughter cell that is away from the basal lamina will undergo differentiation. The first set of differentiation forms the spinous layer. Now, you don't have to remember the names of these layers, but what you do have to remember is as the new cells form and they undergo differentiation, they keep pushing the older cells towards the surface. So once the spinous layer is formed, the cells undergo further differentiation to form a granular layer. Finally, the keratinocytes are formed in the stratum corneum layer. And when they reach the surface, these keratinocytes actually are no longer alive, and hence they are dead flattened sacs, and they are constantly shed, and hence the skin undergoes constant renewal. In addition to skin, the ectoderm can form appendages that include hair, teeth, mammary glands, sweat glands. In the case of birds, feathers. In the case of reptiles, scales, so on and so forth. These ectodermal appendages arise from epidermal placodes. And as we learned earlier, placodes are nothing but local thickening of epithelium. Now remember, epithelium is basically a sheet of cells that are tightly bound to each other. And so we can find epithelium in the ectoderm germ layer, the mesoderm germ layer, and even the endoderm germ layer. However, in the case of ectodermal appendages, they arise from epidermal placodes. Now, these placodes are formed by interactions between the mesenchyme and the ectodermal epithelium. Once again, just to remind you, mesenchyme is when the epithelial cells undergo epithelial mesenchymal transition, and these cells, the mesenchyme cells, are not attached to each other as much. They are more loosely adhered cells. So in the figure that is shown in the blue box, we can see the formation of the placodes, which is basically the local thickening of epithelium. In this case, we are looking at placodes formed for hair formation, tooth formation, and mammary gland formation. And the color coding of the figure is shown in the legend below, where we can see where the epithelium layer is, where the mesenchymal region is, and where the condensed mesenchyme is, which is basically where mesenchymal cells tend to congregate. Now, after placode formation occurs, the cells of the placode will contract and intercalate into the underlying mesenchyme. And this is called as the bud stage, where we can see the epithelium cells that are part of the placode actually go into the mesenchyme. Then several interactions occur between the epithelium and the mesenchyme that ultimately results in the formation of these appendages, like hair or teeth or the mammary glands. During the formation of the ectodermal appendages, the mesenchyme plays a very important role. It condenses at the placode and plays an important role in the induction process. So in the case of some of the appendages like hair, mammary gland, and tooth follicles, the condensed mesenchyme actually provides the epithelium with the signals to form the appendages. So it is the mesenchyme that provides the inductive signals that makes the epithelium undergo differentiation to form the actual appendage structures. The condensed mesenchyme can induce appendage formation when it is transplanted to other types of epithelium that normally would not form those appendages. When this transplantation occurs, we can see those epithelium being able to form the different ectodermal appendages like hair and mammary glands. So just to give an example, let us look at the figure where we're seeing the formation of teeth. Now in this case, we see the formation of the ectoderm and the mesenchyme that is present, and we can see the formation of the dental placode that is shown in the picture that's in the blue box. As development progresses, we can see the bud stage where the ectoderm is going or intercalating into the mesenchyme. And then this is where the mesenchyme is going to start playing a role in inducing changes in the ectoderm. So as development progresses, the mesenchyme condenses and gives signals to the ectoderm and allows the ectoderm to start developing, and ultimately we get the formation of a tooth. Now we look at the various signaling pathways that are involved in the formation of these ectodermal appendages. Literally all the pathways that we have studied play a role. So we can see the actions of Wnt, FGF, TGF-beta, hedgehog, and a new one called ectodisplacin. And all these signaling pathways play a role. So this is shown in the figure when we look at the lower end, where we can see at each step all the different molecules that are involved. And hence the formation of these ectodermal appendages is a very complex process that requires a lot of signaling to occur. Now many ectodermal appendages have regenerative properties. So we all know that if you cut your hair, it grows back. Skin is constantly shed. Teeth can be replaced, so on and so forth. So the ectodermal appendages, they generate or retain stem cells that allow the regrowth of these different structures. So we're going to look at a few examples. For example, in the case of fish and reptiles, they can actually regenerate teeth throughout their lifetime. However, humans cannot. So in the case of humans, we lose our baby teeth or milk teeth, but once we get our adult teeth, if we lose them, we cannot regenerate our teeth back. So the reason that we can regenerate adult teeth is because the humans lose the dental lamina, which has the stem cells that would allow for the regeneration of teeth. In the case of fish and reptiles, that dental lamina still exists, and hence the stem cells are there that can regenerate the teeth. In the case of humans, we all know that we can regenerate hair. Now, when we look at hair, there are different cycles involved in hair growth. The first process is the growth, which is the anagen cycle. Then comes regression, which is the catagen cycle. And then we have the resting cycle, which is called telogen, and the regrowth cycle. Now, we all know that in the case of our bodies, there are certain areas where the hair grows constantly and hence has a very extended growth period, like the hair that is there on our head. However, other parts of our body can make hair that basically have shorter growth cycles, and hence they grow for a span of probably a week or so and then they stop growing. An example of this will be hair that's present on our hands or our feet or even in our armpits. However, when this hair is removed, then we can see the hair growing back again in these regions. And this is simply because the hair follicles enter into the regrowth stage because the stem cells that are present there get activated and are able to regenerate hair. Thus, we can see that stem cells play an important role in the regenerative properties of these ectodermal appendages. So with this, we've come to the end of the part of ectodermal plaque codes and different ectodermal appendages.